Welcome to your USMLE Step 1 review on biochemistry molecular biology, starting with the structure and regulation of chromatin and the epigenetic mechanisms that control gene expression. First, let's talk chromatin structure. DNA in our cells spans about 2 meters long, yet it fits into a microscopic nucleus. That's thanks to histone proteins, which DNA wraps around to form nucleosomes, often described as beads on a string. These nucleosomes then coil to form chromatin, the condensed form of DNA. Chromatin exists in two key forms: heterochromatin. This is tightly packed and transcriptionally inactive. It appears dark under the microscope and is rich in AT base pairs. Think heterochromatin equals hard equals gene off. Euchromatin loosely packed and transcriptionally active. This appears lighter under the microscope, allowing for gene expression. Mnemonic euchromatin equals expressed. So chromatin structure directly impacts whether genes are turned on or off, but that's just the start. Next, we dive into epigenetic modifications. Chemical changes that affect how genes are expressed without altering the DNA sequence. First up, DNA methylation. Here, methyl groups, which are CH3, are added to cytosine residues, typically in CpG islands. The effect? Gene silencing. DNA becomes less accessible to transcription factors. Mnemonic DNA methylation equals muted genes. This process is crucial in X inactivation, genomic imprinting, and tumor suppression. Next, histone modifications, which change how tightly DNA winds around histones. Histone methylation can either activate or repress transcription depending on which histone tail and which amino acid is modified. It's not always silencing. Remember, histone methylation may increase or may silence. Then there's histone acetylation, a key player in gene activation. Acetyl groups added to lysine residues neutralize the histone's positive charge, loosening DNA-histone interaction. This opens the chromatin, and transcription is turned on. Mnemonic acetylation equals active. On the flip side, histone deacetylation removes those acetyl groups, tightening the chromatin and silencing genes. Mnemonic deacetylation means DNA is dead silent. Finally, let's go back to the basics: nucleotides, the building blocks of DNA and RNA. Each nucleotide consists of a nitrogenous base, which can be adenine, thymine, cytosine, guanine, or uracil, a pentose sugar, which is ribose in RNA, and deoxyribose in DNA, and a phosphate group. Purines include adenine and guanine, which have 2 rings, remembered as pure as gold. Pyrimidines, on the other hand, include cytosine, thymine, and uracil, which have 1 ring. Think of it as cut the PY. When it comes to base pairing rules, adenine pairs with thymine through 2 hydrogen bonds, while guanine pairs with cytosine through 3 hydrogen bonds, making it a stronger bond. That's your high-yield review of chromatin structure, epigenetic regulation, and nucleotide basics for USMLE Step 1 Biochemistry. Don't forget to review enzymes involved like DNMTs or DNA methyltransferases, HDACs, which are histone deacetylases, and HATs, histone acetyltransferases. Like, follow and save this video for your next review session. You've got this, Doctor to be,